All right, well, my job is really to kind of wrap things up, and I was going to do that by trying to give you a somewhat speculative picture of the future and where all this could be leading us, because you've spent, this week digging in some detail into some of the tools that are available to facilitate integration of genomics into your research. What I want to do in the next few minutes is kind of ask the question why. Where could this be taking all of us? Before I get too far with it, let me take a moment to, first of all, thank you for participating in the course during this past week, and also to remind you to provide feedback. There'll be a way of doing that. And we've given this course now maybe for 5 years, so we've grown it a lot based on the feedback that we've gotten, but there's still plenty of room, we know, for improving the course. So please let us know what worked well for you and what could work better. So you often hear the idea that genomic medicine is going to transform things in a dramatic way, and sometimes some pretty grandiose estimations of how quickly physicians and other providers will be inundated. But in fact, if you look back now 10 plus years since the genome was sequenced, I don't think you could honestly say that medicine has been inundated. It's certainly been incorporating genomics. And I think it's interesting to look at this, which is referred to as the hype curve. And it isn't specific to genetics, but it's pretty well applicable. Namely that some impressive technology accomplishment, in this case the sequencing of the genome, happens, that it sets up very high expectations about all the great things that will happen as a consequence. And sure enough, soon after the genome was sequenced, there was tremendous sort of hype about all the wonderful things that would take place. And then people realize that things aren't as simple as they were made to seem, and that a lot of the hype is not necessarily realistic, which quickly leads you down to what some call the trough of disillusionment, that this isn't what it was meant to seem to be. And then there's, following that, a slope of enlightenment during which you start realizing how to really do it, and eventually you reach a plateau of productivity, probably not as grandiose as what was projected, but nor as negative as what was feared. And I think you'll find that we're at different places on this curve in genomics for different things. I think there are a few things that have actually already gotten to this plateau of productivity. I'd say there are many more that are climbing this slope of enlightenment. Hopefully we're not stuck down here or, in this kind of precarious position up here. But I think it's a good way to conceptualize. And your job, if you're in the clinical research business, is to help us move up this slope of enlightenment. But if you try to project forward a few, years, maybe decades, as to where it's all leading, I think one way of conceptualizing it is to do a little thought experiment, and that's what I'll ask you to do for this next few minutes. And in particular, we're going to follow a made-up family. Laura is the one that we're going to pay most attention to, but her two siblings, Abby and Seth, to some extent as well. And follow Laura from birth to advanced age in her 60s, as if the question comes up, what can genomic medicine do for her at that point in time? But the one thing about this, which is anomalous, is I'm not going to try to project 60 years from now what genomic medicine is going to be able to do. So at each point in her life, birth, childhood, young adulthood, advanced age, it's going to be as if it were today, which is obviously not how it's really going to play out. But it does give you a way of kind of conceptualizing where we are, and maybe at least not 60 years in the future, but in the foreseeable future, where we're perhaps going. And one thing I'll apologize for is that, as you're going to see in a minute, Laura is a, I guess probably a white middle class individual, and I'm not trying to suggest that that's who will benefit from this. It's just didn't make sense for me to change her ethnicity every time she grew up a little bit. So we had to stick with something. So we're going to consider these 6 epochs: newborn screening when she's born, diagnosis in childhood, preconceptional screening when she's planning a family, prenatal diagnosis when she's pregnant, pre-symptomatic testing when she's a bit older, and predispositional testing when she's significantly older. So we start when she's born, and everybody in the US and throughout much of the world will have some blood drawn, usually from a heel stick at birth, goes to a state newborn screening lab, and really nothing much else is heard. Her parents are just said, don't worry about it, it's a routine test. They never hear more about it. Nobody asks for their permission to draw this blood, it just gets done. And this no doubt happened, I suspect, to most people in this room. It would happen if you have children. This is the blood is taken from the heel stick, blotted onto a piece of filter paper. These days, in most parts of the US, and I suspect around the world, the core test done is tandem mass spectrometry, which is, in essence, you dissociate molecules in the blood, do 2 rounds of mass spectrometry, and at the end you analyze the data to see what metabolites are present. And it really began in the 1960s, so 50-odd years ago, with phenylketonuria, which you may be familiar with, PKU, is a metabolic disorder where there's an inability to break down the amino acid phenylalanine, and the amino acid itself and some of its other breakdown products are toxic to the developing nervous system. And if the baby can't break these down, it damages the nervous system, causing irreversible brain damage, developmental disability, and seizures as an outcome. And why it's important to detect it early is if you do realize that the baby is affected, you can restrict phenylalanine intake in the diet, which is not that difficult to do, and the outcome is essentially normal development. Problem is, if you wait for that to become evident clinically, if you take a healthy baby or apparently healthy baby who has PKU and you wait for the neurologic symptoms to begin, which is the way you would otherwise make this diagnosis, it's too late. The brain's already been damaged and you can't reverse that. So the theory was, and is, that if you make the diagnosis in the newborn period on a biochemical test, then you quickly change the baby's diet to a phenylalanine restricted diet, and they never do develop neurological damage, and indeed it really saves them from a lifetime of neurological disability. And this was the first, but not the last. Now there are about 30 some odd tests that are done. And the PKU test at first was done by an exceedingly low technology approach, one that is now obsolete, but it was important because it proved that you could do it cheaply and reproducibly, and that's what made it possible. So now the idea is you screen everybody at birth for this. It's a 1 in 10,000 disorder, so it's not a priori that likely that any one baby will be positive, but for those that are, it's essentially almost life-saving in a sense, if not saving their physical life, certainly it is saving their brain development. And as a consequence, they also do it without informed consent, because the feeling was that what would be a rationale for a parent to say, I don't want to know if my baby has PKU, and there's a concern that they might say no because they don't want their baby disturbed for a blood draw, not realizing what a devastating disorder they could be missing. So that's been the story for the last 50 years. You could ask the question, would it be more efficient to do genomic sequencing than to do the tandem mass spectrometry? Because the number of disorders you could diagnose that way would be far, far more. And the answer is yes, you could. Everything that's detectable on the mass spectrometry would be detectable on genomic sequencing. But it's a bit more complicated because not only will you detect these things which are known to be actionable at the time when the diagnosis is made, you'd also be detecting a lot of things that you either can't do anything about or which might not actually become a problem until years and years downstream. So you could ask, do you want to know about a baby's risk of Alzheimer's disease, which you could determine from the genome sequence? And of course, it would be decades before that would ever happen, if it was going to happen at all. And secondly, you can't do anything about it with current technology. Or would you want to know about their risk of breast cancer, which you could know from doing a BRCA test, as I mentioned a little while ago. But babies don't get breast cancer, and so most people feel it's appropriate to wait until they're old enough to understand the risks and benefits of testing and make an informed decision at that time, and not to essentially foist it on a baby. So it's a complicated area as to whether it should or shouldn't be done. There actually are several research studies currently underway looking at the efficacy of genomic sequencing in the newborn period and whether it is appropriate to be offering testing at that point in time for things that either immediately will become apparent or might only become apparent many decades later. All right, well, next, Laura is 3 years old, and her brother is just a few years older and has been having developmental problems, and he's identified as having autism spectrum disorder. And you heard from Fadi Mikhail earlier in the week about microarray, cytogenomic microarray testing, where now you can detect copy number changes that are exceedingly small. This particular one shown down here is way below the resolution of the light microscope. So if you looked at the chromosome physically, you wouldn't see anything. But with the array CGH, you can detect these differences. And it's especially useful in autism spectrum disorder, where there aren't that many well-defined genetic disorders or clues to tell you who is likely to have their problems on a genetic basis and who isn't. But by doing this, now the pickup rate, I'm not sure what number Fadi quoted, but it's between 15 and 20% for autism spectrum disorder and developmental disability, which is a much higher rate than was the case previously doing chromosomal analysis. I would say, if you think back to that hype curve, this is definitely on the plateau of productivity. It's a routine test we do all the time and has really proved itself to be extremely useful clinically. It will undoubtedly still evolve and be refined, and technologically will change. I wouldn't assume we'll still be doing it the same way in 10 years as we do now. But the use of genomics as a tool for picking up copy number variation is now quite well established. But the next sort of iteration of this is in genome sequencing. And you heard in some detail from Mike Crowley about the technologies, which collectively are described as next generation sequencing or sometimes as massively parallel sequencing. This is a curve. I don't recall seeing it in his slide set, but it's a tracking of the cost of sequencing from the National Human Genome Research Institute based on data they collect, since they actually support genome centers to do sequencing. So they know how much they're putting into this and how much sequence is being generated. So in 2001, it cost roughly $100 million to sequence a genome. And between then and 2007, it had come down about tenfold in price, which is a really impressive economy. And you'll notice, by the way, it's a logarithmic scale on the y-axis, that the cost more or less followed Moore's law, which is the law that says that the density of transistors you can put on a computer chip doubles every 18 months. And that's why the computers in your pocket are more powerful than what filled a room 30 years ago. And that seemed like a pretty impressive thing. It then really plummeted, and that was when the next generation sequencing. So instead of sequencing just kind of systematically across a genome, you could sequence simultaneously in small fragments, and allowed you to be reducing the cost and increasing the efficiency many orders of magnitude. So by about 2012 or so, the cost of a genome came down to under $10,000, and then it jumped down again, and now it's really hovering just over $1,000. Now that's the cost of the genome sequencing. And you know now that that's only part of the process, right? That establishing the sequence is only the first step. You still have to analyze it. And the analysis, which David Crossman talked to you about, is non-trivial. And that's where the fact that genome sequencing has exceeded Moore's law is a little bit of a double-edged sword, because we need computers to analyze the data. And in fact, they are now the rate limiting step in getting data out. I used to joke that we were headed towards a $1,000 genome coupled with a $1 million interpretation. And I think that was a good soundbite, but it's not really true, because the interpretation costs are coming down too, but not as quickly as the actual sequencing costs. So it's made a big difference in curtailing what sometimes is called the diagnostic odyssey. And this is back to what I talked about in the first hour, the Undiagnosed Diseases program. You have these people that go from doctor to doctor trying to find a diagnosis, never quite getting to the endpoint that they want, and often generating a great deal of frustration and cost. And the question is, can we do better? And I already showed you in the previous talk one example of a diagnosis we made. Here's another. It's a young child with a seizure disorder and intellectual disability and cerebellar hypoplasia as well as brainstem hypoplasia. Sequencing got done on the trio of him and his parents. He had a pathogenic mutation in the gene SPTAN1, and he also had a second one in another gene called SETD5, and neither parent had either one of these. And we can establish that they were truly the biological parents. So these are two new mutations. This is a picture from a paper on a SPTAN mutation, also with brainstem and cerebellar hypoplasia. And we thought that was a pretty good match, plus the variant in question is one that's been reported before to cause this early infantile encephalopathy. The SETD5 is in a region where there's a deletion in intellectual disability, and we're not really sure what it's adding. But by now you shouldn't be shocked to hear of somebody with two independent mutations, because remember that every time a germ cell is made, there are multiple new mutations. And it's turning out to be more common than we would have guessed to have a person who ends up with more than one distinct pathogenic mutation. And presumably the phenotype becomes a kind of amalgam of what either one of them would have produced individually. So if you're analyzing an individual by sequencing and you find something that looks like it's pathogenic and maybe explains part but not all of the phenotype, don't stop looking, because there could be a second pathogenic mutation lurking within the genome. Well, I alluded to this also in the last talk, but I did so in the context of screening. But a point that needs to be borne in mind when you're doing diagnostic sequencing is you're looking at what can end up being the whole genome. And although you're focusing on what could be causing my patient's problems, along the way, you can find variants that might not be causing that patient's problems, but still could be pathogenic. So a few years ago, the American College of Medical Genetics and Genomics addressed the question, what would be appropriate to do in looking for secondary or incidental findings, things that were not the answer to the question, but could actually be medically significant. So they said that mutations on a list, which I'll show you in a minute, should be reported regardless of the age of the patient, that the labs should seek and report specific mutations on the list. There needs to be pre- and post-test counseling. And then the last thing was that patients could, if they want to, opt out of learning about incidental findings. So that was not part of the original recommendation. It stopped at the third bullet there. Because the thinking was, let's say you're doing a genome sequence for intellectual disability, but you happen to stumble on a variant that predicts cardiac arrhythmia and the risk of sudden death, just for example. So you could say, why wouldn't you want to know that you're at risk of sudden death if there's something you can do about it, which there is? for example, put in a pacemaker. And so the thinking at the time was, these are things that are just too important to ignore. There was a lot of pushback from the community on that it was, there are people who might have a good reason not to want to know these things. So after quite a bit of public discussion, the opt-out sort of option was put forward, and that's now what's done standardly. Anecdotally, very few people opt out, and why they might, I guess, is just personal reasons. The metaphor that we've used for this is if you have a chest x-ray, let's say you're in a car accident and you've bruised a rib and you want to know if the rib is broken, they might do a chest x-ray to see if that's the case. And along the way, if the radiologist is reading the image and happens to see a shadow in your lungs, which has nothing to do with your injury but indicates the risk of a lung cancer you didn't know you had, would you want to know about that? And nobody signs a consent when you're getting a chest x-ray that says, I only want to know if my rib is broken. I'm not interested in anything else. As a matter of fact, the opposite happens, which is if the radiologist doesn't report it, and 5 years later you develop signs of lung cancer, and somebody thinks to go back to your previous x-rays and they happen to see a shadow that in retrospect was the first sign of the lung cancer, the radiologist actually could be liable for having missed it. So we didn't want genetics to get into the same boat. But anyway, there is now an opt-out clause, which is rarely exercised, but is on there. You already seen this list. This is actually an older version of the one I showed before, but it consists of predisposition to cancer. I think I've already gone through it, the aortic abnormalities of the heart, high cholesterol, and malignant hyperthermia. You might ask, why do hypercholesterolemia? Just test the cholesterol. And it turns out that there are individuals that have variants, for example, in the LDL receptor, that are at risk of hypercholesterolemia but haven't yet developed it in a clinically significant way that would show up on a blood test. So it actually could be an early predictor. All right, moving on. Laura is now married, and she and her husband looking to start a family. So they meet to get counseling about carrier risks, and they're offered testing among other things for cystic fibrosis. It's a disorder you're probably familiar with, a recessive disorder. It mainly affects the lungs, causes thickened secretions and chronic lung disease. In many people, it also causes malabsorption because of blockage of the pancreatic duct. And the pathophysiology is essentially thickened secretions. Well, in fact, for the past several decades, carrier screening has been offered mostly on a basis of ethnicity. So if you're of Ashkenazi Jewish descent, Eastern European Jewish, Tay-Sachs disease is much higher than in the general population. Cystic fibrosis is, and a variety of others. The list I've seen most recently has 19 different disorders that get screened for. If you're of African, Asian, or Mediterranean descent, various hemoglobinopathies are prevalent. And then cystic fibrosis, although it is most common in the Northern European ancestry population, can be seen anywhere, as well as spinal muscular atrophy. This is obviously impossible to read, but it's a company that offers carrier testing now for more than 100 different disorders. And they're done on a pan-ethnic basis, partly because a lot of people don't know their full ancestry anymore. People are increasingly a mixture of all different ancestries. They may not realize what their true ancestry is. And secondly, although some of these disorders are much rarer in some ethnicities than in others, the testing is now so simple it doesn't really matter to add another test to a panel, such as like a microarray genomic screening array like I talked about in the last hour. The incremental cost of putting it on there is so inexpensive that even though your risk may be low, that you and your partner are both carriers, which conveys a 25% risk to an offspring, even though it's a low risk, the impact can be very high and the cost of doing it essentially zero. So they're now putting a much larger panel of things on these tests than had been the case previously. So you could ask here too, would it be better to do genome sequencing than doing a test one at a time for each of these. And here was a paper where they didn't actually do full genome sequencing with analysis of all the genome. They analyzed 450 genes known to cause severe childhood recessive disorders. And they asked the question of all the individuals, how many people were carriers for none up to 9 of these different traits. And there were a few, but not many, who weren't a carrier for anything. And there were a few who were carriers for as many as 9. Most people in the mode here is 3, were carriers for several different things, and in some cases, quite a few. And I make the point, actually, that if you look for enough traits, every one of us is a carrier for something, almost 100%. And that would mean that if we're going to do it on this scale, we're going to need to have the counseling resources to explain to people what that means and what they can do about it. Remember, this only matters if you and your partner are carriers for the same thing, and then you have a risk to your offspring. All right, well, they are found to be cystic fibrosis carriers, and they're offered, therefore, prenatal testing. And the fetus turns out to be a carrier, but not a homozygote, so the fetus would be unaffected with the condition. So prenatal diagnosis also dates back to the 60s. Amniocentesis, the original approach, done between usually 15 and 18 weeks of pregnancy, drawing some amniotic fluid from the amniotic sac, which contains fetal cells, skin cells, and bladder cells mainly, because amniotic fluid is basically fetal urine. There are cells in there that can be analyzed genetically, can be used as a source of dividing cells for chromosome analysis. You can get an earlier result by doing chorionic villus sampling, inserting a catheter through the cervix or sometimes transabdominally, and removing a very tiny bit of fetal placenta, which is fetal tissue, and then can be tested. And nowadays, you can even do pre-implantation diagnosis. You allow in vitro fertilization, harvest eggs, fertilize them in vitro, allow the embryos to reach the 8-cell stage. At that time, every cell is capable of forming its own embryo, and you can remove one, and it does no harm to the embryo because they haven't differentiated yet. And you remove one cell, you do a single-cell genomic assay, and you only implant back into the mother those embryos found not to carry the mutation that they're at risk for, and avoid the birth of an affected child. It's a pretty expensive and complicated approach, but it has gotten some real interest. Yeah.
So there's a lot of people doing IVF for reproductive issues. They, for sperm and egg donors, do they pre-screen that for your status for things like this, or is that a wait until there's an actual embryo? Or
Yeah, so there is some effort. I think it depends a little on who's doing it, but yeah, there is some effort to screen for the kind of typical things. So you definitely would have the ability to know about carrier status for some of these things. The donors, I think, are also expected to reveal any particular condition, but it doesn't always work the way you might hope. There's a case that our lab got involved in because we do neurofibromatosis testing. So it turned out, and this is a published case, so it's public knowledge, that any given sperm donor, the sperm can be disseminated to lots of different banks, it turns out, and a whole bunch of children were being born, I think more than a dozen, who had neurofibromatosis with a big gene deletion as the underlying mutation, which happens to be a pretty severe form of NF. And it was realized that it was exactly the same mutation, and they were all the product of artificial insemination. And it got traced back to one person, and it turned out he was a mosaic. He didn't know he had it, but he had, in retrospect, some signs that were subtle, but you could determine. And his sperm contained a significant proportion with this deletion, and that caused a good number of people to be affected. And it wasn't negligence, really. It was that he didn't realize he was affected with this, and turned out he was. So yeah, you can, but it's pretty hard to pick up everything. I think it is, although I'm not an expert on the IVF. Well, one of the questions is, does next-gen sequencing have a role in prenatal testing? And in fact, now it does, and it's pretty increasingly routine. Oops. So it turns out that during pregnancy, some fetal DNA is released from normally degenerating placental cells mainly into the maternal blood, where it circulates for a while and then gets cleared. Truth is, we all have some DNA in our bloodstream that is derived from degenerating cells, and it similarly lasts a little while and it gets cleared from the circulation. In a pregnant woman, 10% of that DNA is of fetal origin. And so you can take a mother's blood sample around 10 or so weeks of gestation, do next generation sequencing, and you're sequencing a mixture of 90% maternal DNA and 10% fetal. You can then, using bioinformatic approaches, count how much contribution of that DNA is chromosomes 13, 18, and 21, the X and the Y. And Down syndrome is due to trisomy 21, an extra copy of 21. If the fetus has trisomy 21, there'll be a minuscule but detectable increase in the proportion of chromosome 21 sequences attributable to the fetal DNA, which has an extra 21. The same could be said for an extra 18 or 13, which are the two other non-sex chromosome trisomies compatible with live birth. So you can now screen for Down syndrome, as well as other trisomies that can be associated with live birth, early in pregnancy in a mother's blood sample with very high detection rate and reliability. And this is now essentially standard practice, and it's performed routinely in those individuals that are concerned about that risk. And it's replacing what are otherwise historical approaches of looking at certain hormone levels in the blood or ultrasound markers. You can take it to its logical extreme. Can you sequence the entire fetal genome from a mother's blood sample? The answer is yes, you can. You can similarly sequence all the DNA in the mother's blood sample. Again, 10% of it is fetal. To do it and figure out which is which, you would have to have previously sequenced the mother and the father, and you can reconstruct the fetal genotype. And it could be used as a way to detect specific mutations that are known to be at risk in the fetus. Theoretically, you could do all the things we've talked about with whole genome sequencing in the fetus. So I'll come back to this point later. How much do you want to know about a fetus in terms of their genetic composition? All right, well, next, what about pre-symptomatic testing? So Laura's now 45, and her older sister has been diagnosed with breast cancer, and it raises the question, could this be a risk for Laura as well? This is a different pedigree, but the idea here is, in this family, this person came for counseling because her sister, as well as a cousin, aunt, and grandmother all had breast cancer. There was another aunt with ovarian cancer and an uncle with prostate cancer, and some cancers on the other side of the family too. We actually did testing for the affected sister and found that she had a BRCA1 mutation. It may surprise you that our patient comes in and we test her sister. And the reason we did that was we needed to know, is this family history accounted for by a BRCA mutation? Because there are many other, not always so easily detectable genes that could account for this. Once we knew that, we could test our patient. She was negative, and that was reassuring because we could say the family mutation, which is accounting for the breast and ovarian cancer, you have not inherited. If we had tested our patient in the first place and she were negative, we wouldn't know if she was negative because the family history is accounted for by something other than BRCA. or that she actually didn't inherit a mutation. So the power would have been a lot less. But this is happening every day now. We have patients coming into our cancer genetics clinic. They're referred, like in this case, for a family history of cancer, or sometimes for a personal history. They developed cancer at a younger age than usual for that cancer type, or they've had bilateral breast cancer or breast and ovarian cancer, things that indicate a risk of a genetic predisposition. But not everybody knows about their family history. As I mentioned in the first hour, that's the rationale for the population screening project that we're doing with AGHI. Now, when all this was introduced in the 90s, some people skeptically said, why bother? You know, you're just telling somebody the name of what they might get with no way of knowing if they will get it. The risk of breast cancer could be upwards of 80% if a person carries a mutation. But could you do anything to intervene? And the answer was probably, and now the answer is yes, definitely. This is showing a study where they did salpingo oophorectomy, removing the ovaries and fallopian tubes in individuals with BRCA. And it turned out to be protective against breast cancer, even though the breasts were being left intact. So each tick on this curve is another diagnosis of cancer. And if you didn't have the surgery, but just surveillance, a high proportion would succumb to cancer, far fewer that had the surgery. So now you can offer surgery, including mastectomy, as well as chemo prevention and surveillance as options. So it's not just academic interest to tell somebody that they've inherited a mutation, but it really changes their care for the better. Mary Claire King, who had discovered the genetic link to breast cancer in the first place, has proposed population screening, and there's been a pilot project of doing that at, in Huntsville, done by Hudson Alpha. So testing, genomic testing of cancer extends beyond germline predisposition. Every cancer cell is genetically abnormal. It starts off as a normal cell, but there is a gradual accumulation of genetic alterations, which little by little push the cell on the pathway to cancer. And the final metastatic cancer is the consequence of an accumulation of multiple mutations that release the cell from normal controls on cell growth. And the question is, if you understood what these genetic changes were, could you devise better approaches to treatment? And now it's becoming increasingly routine to sequence the tumor, in some cases comparing it to the normal sequence, although in other cases just looking at the tumor alone, and trying to catalog all the variants that have accumulated in that tumor, and then, if possible, matching them to particular drugs and optimizing treatment. I will say we have more knowledge of the genetic signature of cancer than we have treatments right now, and I think you heard about this in some detail from Sarah Cooper yesterday. The idea of targeted therapeutics, though, is not confined to cancer. Tuberous sclerosis complex is a genetic disorder where you get developmental disability, seizures, abnormalities in the brain, tumors that are benign but can cause kidney failure in the kidney, variety of other tumor types. Here's one example of a benign but potentially dangerous tumor in the brain. Dangerous because it obstructs spinal fluid flow, and here is an example of a very abnormal kidney, or pair of kidneys actually. So the tuberous sclerosis complex is a dominant, and it's due to mutation in either of two genes, but the two genes encode respectively a protein called hamartin or tuberin, and they complex to form a unit which inhibits another protein called mTOR. So mTOR was originally discovered in yeast. It's involved in control of cell growth, and it was actually discovered because it interacts with an antibiotic called rapamycin. And so they named it target of rapamycin. And it was then discovered that the tuberous sclerosis genes regulate mTOR. And when they're knocked out, rapamycin, the target of rapamycin, is hyperactive. So this turned out to be a particularly fortunate situation because the name of the drug that you might want to test for efficacy is written on the label for the protein. if it's a target of rapamycin, people thought, what about trying rapamycin? And sure enough, when they did, these tumors in the brain or the kidney would shrink. And it produced a clinical trial which resulted in an FDA-approved treatment, which is the everolimus, is a rapamycin-like drug that's a little easier to tolerate and is now used routinely in treating people with tuberous sclerosis. Another example, cystic fibrosis, I alluded to a few minutes ago. It's a calcium channel, chloride channel in the cell membrane. Chloride is pumped out of the cell, water passively follows. When that fails, because either the protein isn't made normally or it's degraded, calcium, I keep saying calcium, chloride can't get out, and the water that normally would follow doesn't do that, and the secretions, whether it's mucus in the lung or pancreatic enzymes, are abnormally thickened, and they just create a cement-like coating, which interferes with lung function and with digestion, among other things. Well, a drug company started exploring, are there compounds that could essentially restore function to the mutated chloride channel? And they found one, which was a chemical that produced a drug now called ivacaftor. And here's a clinical trial that shows great improvement in lung function compared to placebo. And it is now part of the armamentarium of treating CF. But the catch is that it only works if you have this specific mutation, G551D, which is not a very common CF mutation. So therefore, you only benefit if you have the exactly right mutation, which isn't going to be very many people. But on the other hand, if you're one of those people, it makes a big difference to you. And now the CF Foundation and the drug companies they work with have been searching for other similar drugs, and they indeed do have one now that interacts with a much more prevalent mutation. But this notion of what you might call mutation-guided therapy has begun to make a big difference in the CF community. Well, finally, Laura is now 60, and she's been basically well. She and her husband learned that it's possible to get direct-to-consumer genomic testing, and this intrigues them. The kind of driving theory here comes back to a point Greg Barsh made on the first day, that we all have some genetic liability towards common disease like diabetes or hypertension, but it's not enough to just cause it. It works together with other genes in the environment to gradually, over time, kind of bring us across this threshold into the disease state. And the question is, if we could figure out what the genetic liability was, and maybe use that information to protect those individuals at risk from the environmental exposures that would trigger disease, or if they develop disease, to treat them better because you understand more thoroughly what causes the phenotype, the outcomes could be improved. So as it began to emerge that there were some associations of particular genetic markers with common disease, a few companies were spawned in the past decade to do testing to see if you carried any of these markers that predicted disease. So actually, before I go to that, only one of them survives, and that's probably familiar to you, called 23andMe. And basically what you can do now is log on to the internet, give them your credit card number. They'll send you a tube you're supposed to spit into, and you send it back, and then a few weeks later they send you an email, and they've meanwhile done genotyping, actually using a very similar thing to the global screening array I talked about, for many hundreds of thousands of single nucleotide polymorphisms. No doctor or other health professional in the loop. And then they introduce you to a website. So I did this a few years ago. And by the way, it's not as easy as you might think to generate that much saliva to fill a tube. I don't know if any of you ever do this. This is what I learned. And I did this before the FDA made them stop returning common disease results. So I found, as one example, that I was at very average risk of type 2 diabetes, pretty similar to the population average. And this was good news. Actually, that's about what I found in general. I was at average risk for everything on their list, which I wasn't unhappy to hear. But I guess I'm not the average consumer, given this is what I do. And you have to read the fine print down here, which I'm not sure everybody does, pointing out that only 26% of the sort of cause of diabetes is accounted for by any gene. And furthermore, the genes they're testing for don't even account for all of that 26%. So they're really chipping away at the edges of genetic risk for diabetes. And furthermore, the populations on which they derive their data may not correspond to the population I come from. So I kind of knew that, and I wasn't going to use this for decision making, although it's kind of a double-edged sword. Somebody could say, if they saw this, that this is great news. I'm immune to type 2 diabetes, so all the information I was provided about losing weight and exercising more is not important to me, because so I can go do as I please, which would be really unfortunate because this is by no means indicating immunity to diabetes. It might indicate your average risk is lower, but not by any stretch zero. So that's the downside of this, which I wouldn't